I move it?
You just go right
This
like that.
one?
That's the next one over.
Okay. So it's very nice to see so many friendly faces again after a few years. I'm happy to be here. I was editor-in-chief from 1988 to 2019. And I just want to say a few things about the journal for those people who are not terribly familiar with our history. Everything you wanted to know in 5 minutes, okay? So the journal was founded in 1920 by William Welch as the American Journal of Hygiene. I still remember. And in the first issue, this is what he said: it's hoped that the American Journal of Hygiene may serve by opening its pages for scientific contribution from such different sciences. He mentioned a lot of sciences, including epidemiology. So actually, epidemiology was the only article published in the first issue of the American Journal of Epidemiology, American Journal of Hygiene. And for many decades after its founding, most papers were focused on infectious disease epidemiology methods, mainly statistical methods and lab-based research. So jump 40 years, because I only have 5 minutes, okay? So you can see 1964, still the infectious diseases predominated with lab-based research findings and almost no chronic non-infectious diseases. And so in 1965, Neil Nathanson was the editor-in-chief. He decided to change the journal's name to American Journal of Epidemiology. And he said, so far as we are aware, there's no journal in the English language which has the word epidemiology in its title. The change in title means that the Board of Editors will welcome an increased number of papers which are significant contribution to the epidemiological method and to the epidemiology of non-infectious disease. Now, remember that even before the change of name, the American Journal of Hygiene published a lot of landmark papers. You know, just 3 examples of those: Frost on the birth cohort analysis, Sartwell on incubation, log normal distribution of incubation periods in a point epidemic, and of course, Bodian, Morgan, and Howe showing that it was possible to immunize people with attenuated virus, laying the groundwork for the development of polio vaccines. So let's go back to the change of name. 5 years later, still infectious diseases predominated. So it takes decades for chronic diseases to take over, and that has been the pattern. Except for a few, there was, when the HIV, there was a few exceptions when the HIV and now the COVID pandemics are going on. So we published landmark papers, methods papers, and we also more recently, we have been probably the main vehicle, one of the main vehicles for modeling of infectious diseases. And Marc Lipsitch, who was one of our associate editors, was really the leader in that area for many years. And Ana Diez, who is the social epidemiology person, she probably recognized that the AJE was also an important vehicle for renewed focus on social epidemiology. And let me just tell you a few of my ideas at what's going to go on, what's going to happen with epidemiology. It's happening now and it's going to happen in the future. Yeah, 1 minute is enough. So my hopes are that more studies based on life course cohorts to better identify early determinants and mediating factors, important for social epidemiology as well. Development of better biomarkers to measure present and past exposures, a better understanding of the potential metabolomics and proteinomics, a growing understanding of the potential limitations of new analytical methods for observational studies. For example, artificial intelligence, we're seeing more and more papers using artificial intelligence methods like machine learning, including those that allow causal inference from observational data. Instrumental variables, the best example. And they actually try to mimic randomized trials. Use of big data with quality assurance and quality control it may be a miracle to happen. And finally, renewal of the recognition of the importance of translational and consequential epidemiology and implementation science, and also recognition that the semantics and choice of model of applied translation epidemiology are not entirely consistent with those of physiological epidemiology. And a simple example is the additive versus the multiplicative model. So that's it. Thank you very much.
Let me see if I know how to do this. Hello, everybody. I'm truly humbled to be here. Thinking about myself, I know that Moyses still remembers everything. I can't remember a lot of things. And so thinking about the progression of where I started and where we are today is, as David, my first paper was in the American Journal of Epidemiology, and is one that is really dear to my heart. And linking the journal to the big episodes and the big changes that are happening across us and makes us think about pandemics. And this is a great day to be thinking about the role of epidemiology in pandemics. So we'll be talking about the future, but let me show you, for those who have not been reading the journal lately and where we're going, and try to think about entice some of you to send some of your papers to the journal. We are trying to continue the legacy of Moyses and the excellence, and try to move in some new directions. So I just a figure that today the journal continues to improve. We are in a positive trajectory, trying to get more papers and more impactful papers. And this goes to this idea that we want to do impactful work. And what means impactful work? It means that common exposures that lead to effects on populations. And so we have been receiving more papers and having more publications. But also, we need to recognize that the world has been changing, and the publications are not only the one that get published in the journal itself, but impact means affecting other ways of that affect our society, which is today social media, includes other aspects of podcasts. And so we're trying to embrace those changes on how to be impactful on those ways. So if you have one of those podcasts or some idea for a podcast or some idea for a tutorial, please send it our way. I also wanted to commemorate the recent passing of Neil Nathanson. Neil was the editor-in-chief, and Moyses mentioned that he changed the name from the Journal of Hygiene to the Journal of Epidemiology. And that was a big change at the time. And one of the things that was magnificent about Neil was that he actually is one of the few authors, I think he's the only author that has published a paper in AJE in every decade since 1950. So, and I also think that his last paper, I hope you get to read it, he wrote a commentary commemorating the 100th anniversary of the journal, wrote a commentary of what was the journal back in those days. And I think that was also his last paper. So it's very fitting to remember Neil in all the contributions that he has made. And I got to meet him a few months ago at Penn, And he's a beloved figure, was a beloved figure, and I really treasure those moments. We also changed the mission statement. And so this is a long list of things to say that we are really looking for impactful work. And what do we mean by impactful work? I mean, common exposures. To be impactful, it needs to be common in the population level. And so those rare etiological studies that rare disease and rare outcomes are not as impactful as it could be for our journal. We are looking for things that are impactful. Also, to celebrate the 100th anniversary, we have done a few things. We commemorated that by republishing critical work in the field. And if you have a chance, there is a landing page of the journal where we talk about landmark papers. And Lori Dean, who I hope she's here online, has a beautiful interview on how do we think about the field as a field. I want to thank this journal work. One of the things that became clear that I wasn't as aware before is that it takes a lot of volunteer work from wonderful people. And so first of all, the organizing committee of the 100th anniversary for the journal, which was David and Ichiro and Polly and Moyses, who put a lot of work into not only today, but the other celebrations that we have for the 100th anniversary. Also, you know, the policy board, the editors-in-chief emeritus and editors in residency, the editors of the journal, the associate editors of the journal, and as important, the staff. The staff of the journal is what gets us going. And today, we wanted to announce a few small things. You know, the journal works based on the willingness of reviewers. So we have an award for the reviewers of the year. So we are proud to announce the reviewers of the year for 2021. And also, we are proud to announce the 10 best papers published in 2021. So I wanted to give a round of applause to both the reviewers and the papers. So. And with that, I think we're going to have a wonderful symposium started by John. Thank you so much for being here, and the floor is yours. Thanks.
Okay, hello everybody, and thank you so much for the invitation to be here. I'm having flashbacks to wandering around here teaching Epi 1 and Epi 751, where actually my principal job was to keep however many, 280 people awake, which involves sort of a high level of entertainment and as much epidemiology as I could sneak in. And those of you who've been here know that. So I am going to ask for your participation today. I may even call on you by name. So I have to say, when I left Hopkins to go to USC, Taddy Dickerson gave me this cartoon, which I'll never forget, sort of universally useful. You can get it from me if you ever leave Baltimore. And, you know, when I got to LA and people would say, well, what do you like better, Los Angeles or Baltimore? And I would just sort of look at them that you can't be asking that question. And, you know, of course, there's pluses and minuses of each. We'll leave that there. And, you know, some things don't change. So this is walking around the Inner Harbor later last night, and it was nice to see that view. Now, I've moved on now. I'm at the Colorado School of Public Health, where we have 3 campuses. We're a collaborative school, which actually leads me to my first quiz, which is coming here. We have to name the mascots of these different places I've been. So Johns Hopkins is what? Fighting Blue Jays, isn't it? Look it up. All right, there you go. Very good. Now, what about USC? Trojans, very good. Boulder? Buffalo, very good. Colorado State? Stumped? Not going to get all the whole. All right. The Rams, Cam the Ram. There's actually a Ralphie the Buffalo who goes out on the field, believe it or not. And the University of Northern Colorado. This is a stumper question. It took me a long time to turn UNC into the University of Northern Colorado. So that's the Bears. And I think the record of the football teams of these might be like 1-15 at best at this point. So, okay, quiz 1. Quiz 2. Who has the second longest tenure as the first longest tenure as Department of Epi chair? Mike Clegg. Who? Leon. Wrong. Al. Oh, no, way off. 5 years. Yeah. David? It's not you either. All right, I'll help you out. It's Frost. It's Frost. I thought that's interesting. I put photos of everybody here. The demographics of the chair and former chairs, I think, will strike you, and I suspect we'll see whenever the next. Well, we'll come to that too, Al. Don't worry. But I think the point actually, it's really interesting that when you look at this department, it has had so few chairs, and I think in part that has helped the stability of the department, its growth and evolution. But it always struck me when I came. There had not been very many chairs before, and certainly I was here 14 years, but Frost holds the record. I also want to acknowledge my debt to Leon Gordis. I still use 2 of his cartoons regularly. Thank God, a panel of experts, which applies to many of the things I do at EPA and elsewhere. And at the start of Epi 1, I think he always showed that You Are Here cartoon, which I was just pontificating at an institution wants to build a new school of public health. And I said, you're here, and appropriately said, good luck. Being here actually made me sort of think back to what I used to say. So I found slides from 2005 when I was teaching the MPH students what we called Epi 1 in the summer. This was the first slide, please. And this was Principles of Epidemiology, the place of epidemiology in improving the health of the public. And here I've sorted through examples where I thought epidemiology had made a difference. We'll come back to this problem of defining epidemiology when I talk about my first lecture in Epi 751. But here we are, and there was a slide, for example, on dealing with epidemics in this lecture appropriately. And here's 4 things to do, and I think those still work. And I found this slide that I'd used 2 years before. We had gone through SARS-1, and you'll remember in SARS-1 we dodged a bullet, luckily, probably because it made people so quick, so sick, so fast that our methods worked. And then tobacco, still, and I've given a talk, in fact, I think for the summer program here, contrasting COVID-19, SARS-CoV-2, and the tobacco epidemic. So still worked. I showed this slide, an article by Ronald Glasser, a physician author, first book on his experience in Vietnam. Public health is often invisible and unremarked. When it works well, when it fails, our neighbors sicken and die. And gee, are we saying this kind of things now? So themes still resonate. On to 2008 and Epi 751, we had a series of lectures, I think in retrospect, they were quite boring, on history, causation, and disease models. And Elizabeth Platts and I were teaching and went through this, and there was this sort of obsessional need to go through all the definitions of epidemiology, which, as Al will point out, is something epidemiologists are still obsessed with. And just to run through a few, I'm not going to ask anyone to do this, but here's Frost. Not merely the doctrine of epidemics, but a science of broader scope, et cetera. And then we wander into this stuff. So this is my McMahon and Pugh book from medical school. You can see well-worn. Epidemiology, the study of the distribution and determinants of disease prevalence in man. The veterinarians would certainly argue with that. And that kind of language remains. So here's Abe Lilienfeld in his book. Here's Leon. I looked at the new edition, and you guys didn't change it. So here it is. Again, if nothing else after this lecture, please change the definition. Thank you. Okay, and when you do that, here's more of this from the dictionary. When you do this, go back to Frost. So this is something else Frost said. He actually said there's a purpose in doing all this. Purpose in view, the purpose in view is always to arrive at a better understanding of its nature, sources, means of spread, and eventually its control, which I thought was pretty well said. So think about doing that. And I think we have to think about how what epidemiology is and what epidemiologists do will change. So I'm going to turn to what I think I'm supposed to talk about, how epidemiology has changed. And start with this. This is from Tony McMichael, a good friend who probably died 10 years ago. And Tony was a big thinker and liked to take on the whole world and how it had changed. One of the first to think about climate change and health, in fact. And if we look, so here's a display of the last 200 years and germ theory and micronutrients and non-infectious diseases, or so-called chronic diseases, social epidemiology, which in some ways has always been with us, and then Tony's big picture looking at systemic change. And a lot of what we're doing now is we build out our causal models and going up more off to the left upstream. That's in fact what we're doing. Over that time, our theories of disease causation have changed. We've gone from miasma, Frost starting to lay that to rest, to germs, bacteria, of course, and on to studying risk factors, genetics, and whatever else comes. So our field is speciated, infectious disease, chronic disease, and more subspecialties and super subspecialties. I mean, you can find people who I used to who studied breast cancer, but they studied hormones in breast cancer or diet in breast cancer, and everything else was just a variable to be controlled for in a model. So, we really have sort of super speciated over time. So what comes next? Now I'm going to give the expanded version of the 5-minute talk that Moyses gave. No kidding. And here we go. So what have we learned? So I have to say this is about my second or third paper published in the American Journal of Epidemiology. It's actually been cited. I don't think maybe not in the last decade, but here it is. And you can see the title is quite scintillating. But I want to spend a minute on the editors in chief because as with the department, there have not been so many editors. And the ones I know best, of course, were George, who has something he wrote, a commentary on a special issue on air pollution in 1978 that I often quote. This is a very complicated issue around what we knew about particulate matter air pollution roughly 45 years ago. And I like that last sentence about judgment in this area depends much more on the art of epidemiology, which I think fits well, the drawing of reasonable conclusions from imperfect data. So if you need a good quote, I think this is one. And then, of course, there's Moyses. And here he is, and here he is again. Now, I show two pictures because the picture on the right did not show Moyses' watch. But the one on the left shows Moyses wearing a watch. I'm not sure what kind it is, but he's looking at his wrist to see if it's the same one. I had many discussions with Moyses about how long someone should be the editor-in-chief of a journal. And he always said, well, I'm thinking in the next 5 years, I might step down. 5, 5, 5, 5. And eventually you get, I guess, to 31. Anyway, so, and I think his caring approach to his job really made a difference. If we look at the journal now, this is from the last 3 years, and you can see that it's truly become an international journal. It's interesting in terms of where the papers are coming from. You can see Harvard up there at the top, the UC system, which is a lot of places, and then Hopkins. The first issue, in fact, all the papers came from Hopkins. There were 5, and they all came from the Johns Hopkins School of Hygiene and public health. The impact factor is interesting. It's not going up, and I think the question is, and the impact factor for epidemiologists, I suspect Enrique knows, is roughly the same as for the AJE. And I don't know, this speaks to something about our field, and that this subspeciation has actually led many papers to go elsewhere. So if somebody's publishing a essentially high-profile paper on the heart, it's likely to go somewhere else. Our field has grown, and in some ways, I would suspect that we should be seeing a growth in the way papers are being cited. As Enrique was showing the question of impact, and I was thinking about Sandro's consequential epidemiology, thinking, how do we really measure what impact our papers are having on the world? And that's a challenge that I know many of us think about, particularly as we get to certain ages and say, gosh, have we done anything? And did that paper really make a difference? And we need a way to measure that. You saw that figure already, so citations are rising in number. Now, over time, and this is just some very simple plots of the titles, chronic disease did rise up. It didn't really make its appearance until somewhat late in sort of the chronic disease transition. And of course, at this school, there was a department of chronic disease until it merged in when Abe Lilienfeld took over the epi and the chronic disease populations. There's a little surge with HIV/AIDS. Okay, and of course, much of that beginning in the early 1980s with some important papers. Health disparities, okay, you can see a rising topic. And then just a quick check on what has happened with COVID, where of course, so much has gone into these online journals and the preprint journals, but here's where things have settled there. I've managed to write a few papers that have COVID in the title, and I think my rejection rate is quite high. So I want to give you a 100-year look now at the AJE. There's that first issue, and Moyses showed the papers from it. There's one that was actually an observational study, the hookworm survey. And then, for example, the one on the left, which would never be done now, some bunny rabbits were exposed to light, and their red cell and white cell counts were followed. Pretty simple, probably maybe hard to get through the Animal Use Committee these days. And then the most recent issue I had when I did this, which was September. And it's actually pretty interesting to look at what the content is. For example, there's a paper on sampling from Google data. Okay, who would have thought? I think 100 years ago, the idea of Google, we'll come back to this, was not there. An old trick, the population attributable fraction, Mort Levin, faculty member here, paper in 1953, and now being applied to estimate how much non-vaccination comes from parental vaccination hesitancy. Curriculum, I think a paper that Anna knows on heterogeneity and spatial inequities in COVID-19. So a very different landscape of papers. Now, I'm going to show you some data on trends. So we've taken the titles for selected years and put them in the word clouds just so you can look at the themes. So here we are in 1921, okay, and you can see what is there. And then we'll go to 22. Hookworm was a favorite, okay, and then you can see 25. Hookworm still hanging around, measles, leishmaniasis, flagellates, infection, obviously, and more. This is 35, okay, 45, there's polio, okay, malaria, plasmodium, influenza. 55, okay, we're still hanging out with infection. Tuberculosis is there. 65, and even in 65, it's still largely focused around infections, which is interesting in terms of what our field was. 75, coronary, if you look, lung, risk, respiratory. So they're starting to see this shift, which in a sense was fairly delayed, I think, in entering into the journal. And then here's 85, and you can see now that shift in what people were talking about and the methods. 95, 2005, 2015, cancer cohort, et cetera. So there's been a real major shift, of course, over time. It was not abrupt, it was gradual, and themes changed. So, and clearly the journal invites has a very mixed picture of papers. 2019 and 2020. And again, you can see social and so on, and 2022, and there's COVID, okay, and making its appearance. So well, luckily it was not me. Yeah, so you can be assured that it was not me, but I had a little help from my friends. And I don't know if you want to see this again, but you get the picture and what the trends are. So, I mean, this is really to then, think about where is epidemiology going in the next 100 years. I mean, clearly things evolved and moved slowly. I mean, if you were around in 1920 or 21, thinking out, it would have been, a challenge to anticipate what is in the next 100 years, but I think we should spend a minute doing that. In November 2020, with my friends Ross Brownson and Graham Colditz at Wash U and Tom Burke, who's sitting in the audience, we wrote what I think was one of the first what's going to happen to the world of public health post-COVID papers. I think we've turned out to be on target in some of the things we wrote, but clearly there's going to be an imprint on public health more broadly and on the field of epidemiology in particular. Now, how long out that imprint will extend, who knows? But when I think about my career, who here used to walk around with boxes of cards? Al, raise your hand. Okay, so some of us walked around with boxes of cards. As a fellow, I was at Harvard, so all the cards said Veritas, truth. Some of us had HP programmable calculators in the book by Ken Rothman and John Boyce, and we could do our own mantle pencils after we took tables off our green bar computer paper from the central computer we were using, the IBM mainframes, shown down there. As a fellow, I used to go to the Harvard Computing Center at midnight because my runs came back much faster. And it was an interesting world. Already sort of the midnight computer geek species had emerged. Now, I was in the Denver airport a couple of years ago, and they were having the last flight of a 747 on United Airlines, a celebration of the 747, which I remember when it came along in about 1970 or so, and that was revolutionized air travel. And now, at least on United, which some of us are prisoners of, they are gone. So things change. So the real question then is, what's the future going to look like? And, you know, if we were to think about the 200th anniversary issue, which will not be on paper, I'm sure, what will it look like? So, you know, some thoughts about where things are going to go. So clearly, data will be key, and the problem of having too much data and more than we can contend with and understand will certainly be with us still, but I think in new ways. You know, when in thinking about public health practice in epidemiology, the old problems are not going to go away. We will still have outbreaks. We will still have clusters. We will still be doing surveillance. We will still need to do all the things that we have been doing. You know, I think we've learned that the new problems may emerge even faster. And I think we've been caught out on any number of things. You know, SARS-CoV-2 being one example. In the world of tobacco control, Juul, electronic cigarettes, came and did their thing far before we knew what was going on. How did I first hear about Juul and its impact? Talking to the mother of a 14-year-old. Okay, I mean, that's probably the kind of surveillance we need. What is it that's hitting our kids that may be a threat to their health? I hope we do better on surveillance for everything, and we make better use and are better connected to healthcare systems themselves. I've worked hard in Colorado on with the state health department and the governor on the pandemic. One thing I've learned is that our fragmented healthcare system is a huge barrier to public health. And even getting the data, even trying to understand hospital admissions, who's going into ICUs, can be a challenge when you have multiple different systems. So we need to do better. And then in academic epidemiology, I'm sure we'll have more new technologies that generate more data. We'll have many studies based in administrative databases and linkages, I'm sure. More omics of every sort. And I think a question to ask is, will there be an identity crisis as to what epidemiology is, in fact, and epidemiologists do? Will we be replaced by computer scientists and AI algorithms in some settings? Possibly true. So if you look at the 200th anniversary issue, which is shown here, I have an advanced copy. We're looking to the next, well, it should be the third 200 years. Does epidemiology have a future? Time for interplanetary epidemiology. Contributions of Moyses Szklo will be there. There's a book review of the great COVID-19 pandemic. Why? You know, if you look at it, John Barry's book, he spoke here, 20 years ago when I was here, when his book came out. That came out about 90 years after the 1918 flu epidemic. So I'll leave you with that. There's this quote that I'm never quite sure where it came from. I've been trying to track this down for years. It's variably attributed to Yogi Berra as our all quotes, and to Niels Bohr, and also to a Danish comedian. But I think this is a good place to end, and thanks. It's very nice to visit. Congratulations to the journal, and I look forward to the 200th anniversary celebration. I actually can make a prediction. I won't be there, so thanks.
Thank you, John. It's nice to have you back and the humor. Our next speaker is Diez-Roux. Anna is a graduate of the Department of Epidemiology, a PhD. She studied with Moyses, correct? Health policy and management. We count you. Yes. Anna is the Dornsife Dean, and she is a distinguished university professor of epidemiology at Drexel University. And it's a real pleasure to have Anna back. And we've been great collaborators for many, many years, but she stole my best doctoral student.
Okay, this is the. So hello, everybody. It's really a pleasure to be here. I really like coming back. It brings back a lot of memories, although of course it's a very different building than the one I was in, but I get a lot of flashbacks, as John was saying, about sitting in a classroom with David, taking your social and behavioral, I can't remember what it was called, but fantastic class, and Moyses, of course, and so many others that I learned so much from here. So thank you for having me. So I'm going to talk a little bit about the social determinants of COVID-19 and reflect a bit on what we saw, what it means, and what some implications or lessons might be for epidemiology and for the future of epidemiology, perhaps. So I'll talk a little bit about the social patterning of COVID-19. This will not be news to you, but I want to say a few things to sort of frame some of the points that I want to touch on later. Talk a little bit about what this means, what drives it, what lessons we can draw from what we saw specifically for epidemiology, and then reflect a little bit on some broader implications for the future. So what we saw, again, this will be no news to you, but a few highlights. First and foremost, of course, these enormous differences that we saw across racialized groups in the United States and also in many other countries. This is cumulative data through September, and particularly for hospitalizations and deaths, 2 to 3 times higher rates. So remarkably strong patterning. This patterning was present not only for race and ethnicity, but also for, oops, this doesn't seem to be advancing. Oh, there's someone's asking for live transcription. I'm not sure what to do. Maybe that's what's blocking it. Let's see. I think it's okay now. Yeah, so of course, this patterning was present not only by race, but also by socioeconomic position. There are a lot less indicators of this. So often we have to use neighborhood level measures. And this is an example, actually a paper that Usama Bilal led, looking at relative rates associated with a neighborhood SES index for a variety of COVID-related outcomes, for incidence, for mortality, also for testing, for positivity. So again, the patterns that with higher rates and lower socioeconomic position groups. This patterning is also emerging for long COVID. These are some data. I'm sorry that the labels on the top are so small, but this is from the NCHS Household Pulse Survey for June through September of 2022. And on the left panel, it's the proportion of people who ever experienced long COVID among those who had COVID by education. The red at the top is the lowest education group. The purple is the highest education group. So very strong patterning in long COVID as well. And on the right, by race, ethnicity, Latinos and African Americans at the top, and then whites further towards. So this pattern is emerging for long COVID as well, although of course there's less data, a lot more complicated to define long COVID and so forth, but emerging too. So not only disease outcomes, but also access to treatments and prevention measures were patterned by social circumstances. This is data on vaccination rates by CDC Neighborhood Vulnerability Index for various cities in the United States. I think this may be from the paper that John mentioned, and you can see on the, again, a patterning by which higher vulnerability is associated with lower complete vaccinations. This was done through September 2021, so during the first 10 or 11 months that vaccines were available. Now, a point that I'll come back to later is that for some cities, you can see the bottom left panel, some cities on the West Coast, the patterning was not as pronounced, and that's interesting. It raises some questions about why that might be. Inequities in treatment too. We are seeing these emerge as well in terms of access to Paxlovid. And you can see at the top the availability of active provider sites for antiviral therapy is higher in areas with high social vulnerability. But on the lower right, you can see that the actual prescription rates, the dispensation rates are actually much lower in the neighborhoods with higher social vulnerability. So clearly, access to treatment is also socially patterned. Now, the pandemic had a number of social consequences itself, consequences of infection and consequences of what we did as a society in response to COVID, like the lockdowns. On the left is results from an interesting study published in PNAS showing the impact of using a cohort study that had a survey right before the pandemic hit, and they did a subsequent survey, and of course showing predictably that the social consequences, housing insecurity, food insecurity, financial insecurity, and employment insecurity, and becoming unemployed, also strongly patterned by social circumstances and race, and even after one controls on the right for pre-existing insecurities or social conditions. Now, of course, this is no surprise. And in fact, the patterns that we are seeing were totally predictable and similar to many things that we have already seen over and over again over the years in public health. This is data from a study in the UK of showing adjusted hazards ratios associated with the Townsend Index, which is an index of deprivation. I remember reading a lot about that index in a cubicle on the 6th floor in the other building. And what you can see is that the patterns for COVID on the left are exactly the same as the patterns for influenza and pneumonia, and are exactly the same as the patterns for cardiovascular disease. So it's quite striking. Even though we know this, it's striking to see how consistent this patterning is. Now, that being said, despite the pervasiveness of this patterning and its predictability, there's also heterogeneity. And I think this is an important point. Here on the top left is a snapshot from a COVID dashboard that we did at Drexel, tracking inequities across cities during the pandemic. And what it shows on the top left, and I'm sorry that the labels are so tiny, but it shows differences between Black people and white people in COVID mortality for different cities. And they're all in the expected direction, higher rates among Black people than white people, but the size of the difference is highly heterogeneous. On the bottom right, there's another figure, and this is from a commentary in the AJE, actually written by Alina Schnakemal and Usama Bilal, tracking the evolution of the pandemic based on the race composition of the counties. And you can see that the peaks occur at different times for counties with different composition. And the reason I show you this is to highlight the fact that depending on when you look at it, you're going to see slightly different race patterning because of the way the pandemic is moving through the population. This is further illustrated by these other data showing age-adjusted deaths by race over the course of the whole pandemic. And once again, although there's a general consistent pattern in terms of the levels for the most part, depending on when you look, they're going to be a little bit different. And similarly for vaccination rates, although overall, we see, certain groups that are always more highly vaccinated than others. There's a crossover, for example, between Latinos and whites towards the end, where the Latino rates became slightly higher than the white rates. So what did we see? Well, we saw predictable social patterning of cases, hospitalization, and deaths. Access to prevention and treatment is also socially patterned. Heterogeneity over time and place, and the social consequences of the pandemic reinforcing prior existing social and health inequities. So what does this tell us as a whole, holistically? Well, to me, what it illustrates, again, is that the causes of health inequities are structural and systemic. And what do I mean specifically by that? These words are used a lot nowadays. I'm a firm believer in being precise in how we use language. So what do we mean exactly by this? What do we mean by structures? And these are different definitions from the dictionary. Structural means relating to the arrangement of and relations between the parts or elements of a complex whole. And systemic means relating to a system, especially as opposed to a particular part. So this means that in order to understand the reasons for this patterning, and which is critically important in terms of understanding how to intervene on it, we have to recognize the role of structure and the role of systems. And this is, not hard to envision for COVID. Let me show you just a very simplified conceptual model. This is class inequality and systemic racism shape a variety of living conditions as well as access to healthcare. Work conditions also impact access to healthcare. These things affect exposure to the virus, pre-existing chronic diseases, access to testing and vaccination, access to healthcare, of course, affects these things as well. Testing affects exposure because of the ability to isolate, et cetera. And these things affect, of course, infection and the extent to which infection evolves into severe disease and death, and so on. And in addition, there are some backwards arrows. There are some reinforcing loops in here, and I'm just highlighting a few. Chronic diseases affect housing and working conditions. Class inequality and systemic racism reinforce each other. And so what this means, what this kind of diagram means, is it illustrates how the causes of health inequities result from multiple intersecting social processes, involve factors at multiple levels, involve long causal chains, life course, and even intergenerational, involve social behavioral healthcare and biology constructs, and really are full of interactions and conditional effects and heterogeneity of effects. And more generally, we're really dealing with dynamic systems that are generating these patterns. Heterogeneous and interdependent units, recursive relationships, nonlinear effects, multiple paths to the same outcome. People get COVID for different reasons. They get heart disease for different reasons. Similar distal causes of multiple different outcomes. The diagram that I showed you, we could put other health outcomes and on the right, and it would be pretty similar in many ways. Oops, I don't know why it's not moving again. Is there something? Keyboard. Okay, the role of history writ large. Use the arrows here. That's another option. Okay, the role of history writ large, and not just life course intergeneration, but also historical processes. And as a consequence, we have emergent patterns that are not easily reducible to independent effects. So let me try this. So all these things result in something that's called policy resistance. And we see this all the time in health inequities, the tendency for interventions to be defeated by the system's response to the intervention itself. The fact that, or maybe we are successful in changing one small piece of it, but then it reappears due to the underlying dynamics that are driving these things. And so as a result, health inequities are big, complex problems. But at the same time, that doesn't mean that we should be paralyzed by this, because in fact, the fact that they are systemic also means that they are modifiable, that they are not natural, that they are not inevitably, that they are a function of the systems that we have created and that we maintain. So, to me, this is in many ways, COVID, the social pattern of COVID illustrated these things in a very stark manner, but this was not new. This is something that we have been talking about in epidemiology for many, many years, but it was a very stark illustration of this that happened very quickly and that acquired incredible public resonance in a way that we had not seen before. But are there specific things that we learned from COVID? And I think there are a few things that I want to highlight. First of all, I think one really, to me, important aspect is that I think COVID illustrated how description and surveillance can be powerful tools to both demonstrate social causation and to motivate action. Because just the fact that we were able to, that these very stark patterns were made visible in a completely different way, not in academic publications, but all over the media, showed how powerful that can be, not just in describing the problem, but also in inferring causes, just from the description, without any fancy causal inference. Despite that, we're behind, right? And in the figure that I showed you before, only 65% of the case records had race ethnicity in the United States. In a rich country, 35% of the case records had no race and ethnicity information attached to them. And similarly, indicators like income and occupation were almost completely absent. But so, to me, this calls for a system for monitoring health inequities that, yes, can be used to target resources, but that can also be used to make the case for social action. Of course, there are a lot of methodological complexities in description, actually very interesting ones. And these are a few examples of papers, a couple of them from the AJE, one paper looking at, should or should we not adjust for place when estimating race differences? What does it mean to do? What is it, just from a descriptive perspective? Or the combination of quantity and qualitative approaches is really cool for descriptive also purposes, a very cool approach called configurational comparative analysis, for example, that can be used to powerfully describe in a more nuanced way social inequities, or even just the distribution of disease. And most recently, it was really nice to see this paper on a framework for descriptive epidemiology, a methodologically rigorous approach to description. So I think this is one interesting lesson from the pandemic. Another interesting lesson, I think, has been the critical roles of work and housing. This is something that I think, to some extent, social epidemiology does not pay as much attention as we should have to. And of course, there have been social epidemiologists who focused on work. Karasek, some of you may remember. I remember learning about this probably in David's class. I remember Jeff Johnson here at Hopkins, from whom I learned a lot about work context and health, but not a lot of attention to work. And I think COVID really highlighted, the role of precarious work, the lack of social protections at work conditions among essential workers as critical drivers, not just of COVID, but probably of many other health outcomes. The fact that occupations are incredibly socially stratified, and therefore they're big contributors to health inequities. Oh, this one's not working. Okay, so these are a couple of papers also from AJE highlighting the same thing. So it's interesting to see this topic of occupation come to the fore. Housing is another domain. I think that got a lot of attention. This is a modeling paper looking at the impact of projecting the impact of evictions on COVID, and showing that evictions potentially, under the assumptions of the model, could impact rates among those evicted and who they want to live with, but also among the general population, which of course makes a lot of sense given transmission dynamics. But also moving beyond, and in a way, moving beyond the usual way in which I think public health is focused on in housing, which has to do with very important exposures like lead, indoor air, mold, but also looking at the impact of housing on birth outcomes, the impact of housing on mental health, for example, in young adults, the impact of housing on health insurance, keeping your health insurance, the multiplicity of ways in which housing can be related to health as well. Lesson 3, don't forget healthcare. I think this is another thing, I think that social, in many ways, folks who focus on the social determinants of health, I think for good reason, have tended to say, well, it's not just about healthcare, there's all this other stuff. And that's really a very important point to make. But at the same time, I think what COVID also illustrated is that healthcare can also be very important. We saw this in the slide I showed you on Paxlovid access. But this is another paper that projected the impact of health insurance, having access to health insurance on COVID outcomes, and suggested that, moving from the current health insurance rate in the US, this is an interesting panel study using states, to full coverage of, if everyone had health insurance, based on their estimates, would have resulted in significant reductions in hospitalization cases and deaths from COVID as well. Lesson 4, I think, that emerges from the COVID experience is the importance of capitalizing on opportunities to evaluate social policies, using, a number of natural experiments, a number of methodologic approaches that many people are writing about these days. These are a few examples looking at eviction moratoriums on COVID-19, variations in relation to COVID, also in relation to mental health, for example, and paid sick leave in relation to emergency department use. And members of our team, led by Alina Schnakemal, has a paper in press, very interesting paper on paid sick leave and vaccination, COVID vaccination as well. So the opportunity to really evaluate policies, something that we don't do enough of. And the 5th lesson, and perhaps the most important lesson, I think, is really embracing and grappling with a more realistic, and by this I mean a more scientifically accurate approach to understanding race differences in health, and which means a focus on racism. Moving beyond race as a variable, recognizing the role of multiple reinforcing processes, recognizing the critical role of life course and history, structures and systems, and practices and policies. All of these things are the ways in which racism affects health. And here I just want to put up this quote from John Powell that I like quite a bit. So he talks about this in the context of evaluation, but he says a systems approach, but it's totally analogous. A systems approach is needed because racial conditions must be seen not simply an outcome of certain attitudes of policies, but as a dynamic, interconnected processes that are part of a larger socioeconomic and political system. And, what does this mean? What does this mean? Well, we have seen, this discussion of many of these very important issues in the American Journal of Epidemiology actually recently. And I think we will see more. So here are a few examples: a paper by Lisa Burrell and Natalie Crawford, paper by Lauren Dean, talking about structural racism and measurement. There's actually a very interesting, I think, discussion going on about some of these issues. Another paper by Paris Atkins Jackson, Jackie Jan, who is a faculty member with us at Drexel as well. So I think a lot of important discussion, and with empirical implications, I think that has been motivated by this recognition that we really need to look at, quote, race in a different way. So these are my 5 lessons from COVID. Description and surveillance as powerful tools, critical roles of work and housing, should not forget healthcare, capitalize on opportunities to evaluate social policies, and a more realistic approach to understanding race differences, focus on racism. And now in the last few minutes that I have, I want to reflect a little bit on some broader implications for epidemiology. And some of these have been sort of foreshadowed by some of the things I was talking about. But the first is the importance of grounding research in theoretically based and systemic conceptual models. I think sometimes, you know, it has happened in the field of epidemiology that we have become, in some cases, more driven by the method than by the question. And so basically we ask the questions based on what the method we know will provide, instead of asking the question based on what the important questions really are. And having these concepts and theories is super important, not just in developing the questions, but also in putting the results together to develop socially useful explanations. And this is a little bit about what Frost says in that quote. There's a quote that he says about putting things together in an ordered chain of facts. And that's really, really critical. To me, that's what epidemiology really is. This is having these kinds of concepts and theories is fundamental to framing and identifying questions, to guiding everything we do, guiding the data that we collect, how we interpret the results. Now, these conceptual models need to balance generality and specificity. We can't have a general model that applies to everything. We need to get specific. That's what science is about, right? So we really need to make these things specific in terms of level, time and history, dynamics, as they relate to a specific question. And these things have to be susceptible to empirical verification or refutation. We need to be able to show there has to be something at risk. We need to be able to show that we're right or that we're wrong, and therefore, you know, something else is going on. My second sort of big picture, kind of broad implication, and this is sort of a favorite topic of mine, is that we really need to think of that there's a continuum of policy relevant questions that we can look at in epidemiology. Questions about distribution, descriptive, purely descriptive, questions about etiology, and questions about interventions. And all of these can inform etiologic understanding and action. You know, I think there has been somewhat of a movement in epidemiology through the, you know, a very important focus on quantitative causal inference that has shifted the discussion into, you know, formulating every causal question as a potential intervention. But that has its drawbacks too. And I think we lose, by going exclusively in that direction, we lose the ability to look at these big fundamental drivers, which cannot always be tightly specified as a single intervention. And this is a quote that I like also about this. There's a counterfactual interventionist notion of causation, of use when one is designing a public policy to intervene and solve a problem, and an historical, or more exactly ideological notion, often of use when one is identifying a problem to solve. So it's both things that we need to think about. So I like to sort of illustrate this with a little diagram. You know, if we think about broad antecedents leading to multiple causal pathways, socioeconomic position is the one example of a broad antecedent. We can further specify that as one characteristic, like education, or we can transform that as we can formulate that as a hypothetical intervention, increasing years of education, a practicable intervention, something we could actually do today, a practical intervention under specific circumstances. And as you can see, you know, if you go up, the causal question becomes broader, and it requires a different approach than a very narrow and tight causal inference approach to answer the question. But as we go down, it becomes much more specific, and it essentially becomes implementation science in many ways. So really, there's a continuum of questions which can all inform ideologic understanding and action. A third sort of broad implication is that I think we need a systems approach to epidemiology. And by this, I do not mean using systems modeling all the time. I mean expanding the conceptual models to recognize the role of structure and systems and dynamics, but then exploring them with different kinds of methods. Multiple types of questions, multiple types of methods. A narrow quantitative causal inference approach is insufficient. And also, what I was talking about before, rethinking the questions. So, developing questions that are about processes rather than about static comparisons, and also allowing ourselves to imagine new interventions that have not yet happened in our world, but that could happen and that could have important implications. I think another related area is how we think about influencing the policy agenda. And this is from a really cool paper that by Carol Weiss, who I believe was a sociologist. She may have been a political scientist, I'm not sure, that is entitled The Many Meanings of Research Utilization. And she outlines the different ways in which research can impact policy. And here are two common ones. She describes different models of how this can happen. So knowledge-driven, when basic research identifies an opportunity for application, applied research tests the practical application, technologies and applications follow. That we're very used to that. Or the one below, problem-driven, a policy problem is identified, a decision has to be made, research is conducted or identified that sheds light on the problem and helps identify the best policy option to solve the problem. But there are a whole bunch of other ways in which research can influence policy. And she describes them here, and I'm not going to go over each of them. They're all interesting, I think. But the one that I want to focus on is this enlightenment way. When the concepts, theories, and basic understandings that science has generated permeate the policymaking process. And for this, this is not only about testing a particular intervention and being able to tell a policymaker, if you do this in your state, this will happen. This is about changing the way in which we think about the drivers of health. And therefore what is possible, what kinds of interventions or policies are possible, feasible, or desirable. It's about changing the mindset. And in order to address the social determinants of health, we're not going to do it, but identifying interventions and policies that work is very important, but we need to address multiple things at the same time. And I think that this is why this enlightenment model is so appropriate. And so when we think about our results, about our research, about the type of research that we do, we need to have a broad approach to thinking about how we can influence the policy agenda. And I think COVID has illustrated this because some basic descriptive statistics on COVID had perhaps more impact than a lot of policy evaluation studies that we could have done. And my fifth broad implication is that we need, in order to really be able to grapple with the complexity of the problems that we have to grapple with in terms of social determinants of health, we need diverse and inclusive teams because they're critical to these goals. They're critical to the formulation of these realistic conceptual models because the experiences of different people, different kinds of people, different histories will enrich the development of those models and make them more useful. And this influences, the questions that we ask, the hypotheses that we develop, allows challenges to the dominant paradigm that we might be working on, where we just sort of stop seeing certain things because we're also used to doing the same thing over and over again to publish our papers, for example, and using the same methods over and over again. Interdisciplinary and diverse methodologies, avoiding disciplinary biases, quantitative and qualitative approaches, it has importance for the collection and interpretation of data and for inferences. So it's really about changing the perspective. Increasing diversity changes the way we look at things and allows us to see reality, I think, more accurately and therefore to do better science. And there's a lot of evidence that diverse teams are better at solving problems as well. This has been demonstrated empirically. Also, the ability to identify interventions and policies that we might not have thought of. What is desirable? What is or should be possible in our society as an intervention or as a policy? policies or interventions outside the box. For all of these things, to be able to even imagine these things, we need diverse perspectives in a research team. For communication dissemination, this is critical as well. For this enlightenment role of evidence, for empowering those historically marginalized through communication. And last but not least, because I think there's also a moral obligation to redress unfairness and past injustices and to increase opportunities and resources to replace exclusion with inclusion. And this has many synergistics and multiplying effects. So these are sort of my broad implications. Writing research in theoretically based and systemic conceptual models, exploring a continuum of policy relevant questions, a systems approach to epidemiology, a broad approach to influencing the policy agenda, and diverse and inclusive teams as critical to our goals. And I'll just finish up with two quotes. As you can see, I like quotes a lot. This is a quote attributed to Noam Chomsky, although there may be other similar, he may be taking it from other folks as well. Science is a bit like the joke about the drunk who's looking under a lamppost for a key that he has lost on the other side of the street because that's where the light is. It has no other choice. So of course, we don't want to be that kind of scientist. We want to look outside. A quote by Forrester, in the complex system, causes are usually found not in prior events, but in the structure and policies of the system. So this is the big challenge for us, and this in epidemiology generally, but in social inequalities in particular, and why we need a broad perspective, multiple methods, diversity, and multiple kinds of questions. So that's it. Thank you. Thank you very much.
So our final speaker is on his way. So we're going to take a break now. I guess we will, I don't know how okay, 1:50. So thank you all. Chance to get some coffee.
No, we have to go down and across, but it's okay.
The mics are.
Yep. So while we wait for Mark, I wanted to start and maybe start the conversation and asking John first a question that maybe you can give us some of your thoughts. When you talk about the next 100 years, I wonder what are your thoughts? And the fact, there are some fields that over time, they remain with the same name and the mission and vision remains the same. And epidemiology may or may not be that field. Are we going to be doing epidemiology? Is it going to be the same discipline? You think it's going to require changing the name of the profession? Or are we going to remain as we are today?
Is this on? That's too bad. Fields come and go, don't they? And, 20 years ago, how many of us were saying data scientist or other terms? So I think we have to acknowledge that nobody's an alchemist anymore. And, perhaps we should understand that there will be people using information about the health of populations. This can come in many different ways to analyze it, ask questions, hopefully answer them and make things different. I mean, I think already there are many people who are doing that who may or may not call themselves epidemiologists. And I think if you look at healthcare systems, for example, as they try and meet their goals of saying that they are doing things to improve, quote, population health, they may be data scientists, data analysts, and others. Perhaps, thinking about Anna's talk, they may not be building models in their head about how the world works when they analyze the data. They probably aren't, in fact, but I bet they're producing reams of reports and lots of graphs. So I think we should think about that. I have these conversations with my colleagues.
Come on in. Good to see you guys. Good to see you. Here, let me just turn this on. Feels like forever. Yeah. So we're seated, and I don't know to what extent you're still plugged in on CTY.
Steve Ganji, you're not on mute.
Request for retention payments, tax payments. And my first instinct was, this doesn't feel right. Particularly knowing what I know, but apparently there have been some folks who have been transitioning out, which I thought, quite frankly, it's a good thing. Yeah.
It's also transcribing.
It just feels still odd for me for us to be thinking about retention payments for people.
So that's probably because we made the decision to defer the annual merit increases, right? Which were scheduled for October 1st. And it made no sense for us to be off schedule from the university.
Right.
And quite frankly, it helps the bottom line.
Yeah.
Sorry.
And where was I? I was actually, the other comment I was going to make, had this conversation, actually, I've had this conversation many times with my colleagues in biostatistics. Moyses Szklo is our chair of biostatistics and informatics. And we've had this conversation many times about whether there are people, so quote, biostatisticians. Our last probably 3 or 4 hires are coming from computer sciences departments and not from sort of usual departments of biostatistics. So I think we have to acknowledge that things will change. And I don't know if anybody else wants to take that one on, but change is coming.
Anybody else wants to comment on are we going to be epidemiologists in 100 years from now, or will be data scientists or any other? And I have a question for you. You know, I'm going to be provocative on purpose. You know, if race or structures or poverty were something that the FDA will evaluate, it will be forbidden, right? There will be actions about it. There will be trials. There will be ways to evaluate interventions to reduce the risk. And in social epidemiology, and especially in issues with racism, we have a lot of really, really good methods and observational data, but we haven't done, in comparison with other fields, enough trials. And I wonder, what are your thoughts about moving to thinking about interventions that will lead to changes in policies? That dream are too hard to do? What are your general thoughts on that?
Is this working? Yeah. Yeah, I mean, I think there are. So I think there are some things, some policies, some potential interventions for which we should do trials, or we should take advantage of natural experiments to evaluate their impacts. But I think with the social determinants of health, with the impacts of racism on health, we're not going to do randomized trials for everything. We couldn't, and we don't need to. And there are other, there's enough evidence to act, I think, without doing randomized trials. And you can say, well, we don't know how to act. Well, that's true, but it is a very complex social problem. And so we need to try things. And there may be things that we need to do as a society for reasons that have nothing to do with health, just for reasons of justice, that we will see will have health consequences. And we don't need trials to do that. We need, as a society, to recognize that we need to make those changes for a variety of reasons, not just because of health, and that we will probably see a health impact. And yeah, we should evaluate that to document it. But I think it's unrealistic and I think naive to be in the mindset that we need, clinical trial evidence or we need specific policy evaluation studies before we can act on the social determinants of health as a society. So I don't know if that totally answers your question, but that's sort of.
No, so let me ask back. So one of the things is that usually when we get from the outside word of people who are not in our field is to say, well, observational data has too many confounders, it's not as reliable as it could be, and how can you do an intervention that it's based on observational data? And we know that that's not the case. There's a really strong evidence from observational data. So what do we need to do to change? Therefore, if we need to continue to do observational studies in a better ways, what do we need to do to communicate that data coming from observational studies could be for evidence generating as good as a randomized trial? What are your thoughts,
Well,
and
I mean,
especially in social epi?
Yeah, I mean, I think that the issue of the utility of observational studies is extends beyond social determinants, right? I mean,
Sure.
the EPA stuff, the use of observational studies and setting, regular standards for air pollution. This came up, they're throwing out all the observational data a few years ago, right? No observational studies were going to be used because they were all hopelessly flawed. So I think, I think it's a question of, and in fact, that quote, who was it? Quote you put up, drawing reasonable conclusions from messy data, right? This is what observational studies are about. And I think we have to have a perspective. And that's what I was trying to illustrate with that table from Carol Weiss, that, in developing policy recommendations or evidence to justify policy, you need a bunch of different kinds of evidence, some of which may be trials, some of which may be observational, some of which may be mechanistic animal studies, some of which may be simply description to say, look, hey, look, the rates are 3 times higher in Blacks and whites. The life expectancy of Black men in this country is 67 years. 67 years. in a rich country. You know, and that in itself should motivate policy discussion. So I think it has to be, we have to think of evidence for policy in a broad way. Narratives change policy. The way we put together stories about what's important to health affects policy too. So it's not a narrow thing. It's a big thing because these are big social issues. So.
Thank you.
Sorry, just to keep you on the spot. The data you showed on the disparities, you could have anticipated those. And as you showed, so I guess the question I would put to all of us, then I'll hand the microphone to you, is should we have done a better job of anticipating, preparing, identifying where those disparities were coming from and being proactive? And at what point do you think we had enough data to see whether it was through workplace approaches, through looking at the gaps in healthcare delivery, surveillance? Did we do things in a timely way in response as these data emerged? Because some of this came along fairly quickly. How do we use what we learned? And so I guess on the anticipatory side, we probably should have done much better, and we didn't. And then did we learn? Well, that's so. Okay, Al. Who should the we have been?
Well, we should have been A and F, but that's not who we are.
Yeah. So there's lots of we's out there, and fair enough, because you know, I mean, if you look at Colorado, we have 54 local health departments that act, you know, on their own, and we have state, and we have many other actors. So I mean, that again goes back to how does this information actually get to those who might make a, you know, might make a difference. But, you know, could we have done better? I'm handing this off, and all of us are we's who are in this auditorium. So I think that's probably a fair way to put it.
Well, I mean, could we have done better? Yeah, obviously, yeah. I mean, I think, but this is I mean, the response to the pandemic, I mean, in general, it was chaotic. I mean, it was completely chaotic. Despite all our investments in pandemic preparedness, and maybe we should, you know, what did we see? We saw chaos. I mean, that's what we saw. And so it was totally predictable that we were going to see the social pattern that we saw. It was totally predictable. Are there things that we could have done? Yes, there are many things that we could have done and we didn't do. But that, I'm not sure it has to do with specifically with social determinants of health. It's more of how we responded in general to the pandemic. I mean, I was on the vaccine, there was the National Academy of Sciences had a vaccine allocation committee convened, you know, it was August right before. And, you know, we developed a set of recommendations as these committees do. And, you know, a key recommendation was allocating vaccine based on neighborhood indicators, socioeconomic indicators. Did that happen? No. In a few places it did happen, actually. Actually, in some of those cities on the West Coast that I showed in that figure that Usama did, they did a little bit of that, and that's maybe why they didn't see as big inequities there. But that, you know, that's interesting. But those recommendations were out there. But then, you know, it happened in a haphazard way. Different jurisdictions decided on different things, and so that all went out the window. But there, you know, there were specific things that we could have done. Absolutely, yeah.
Okay. So what I would say is, seeing that Marc is here, is that a good time to get Marc to start, and then we'll continue the conversation? Is that good? Okay. Well.
And we have an intro slide for him. So we have that, and then I'll hold his. That while when you're introducing him, and then I'll hold his slides for you.
Good. So our third speaker today is Marc Lipsitch. He's a professor of epidemiology at the Chan Harvard School of Public Health, and he's director of the Center for Communicable Diseases Dynamics. And he gives a very exciting talk. So Marc, you're up.
Thank you. And Thanks to AJE and to David for the invitation. And I apologize for being late. I don't believe in showing up at this kind of thing and flying in and flying out, but this is my own personal conflict between medicine and public health. I had a doctor's appointment that I wasn't going to get another one if I didn't take it, so I took it. Apologies for that, but it sounds as though the discussion has already touched on a lot of things that would be fun to think about in the context of what I want to say. I think I'll just start by saying a subtext of what I prepared, which is not very much emphasized in the way I put it together, but I think is worth thinking about, is that virtually everything we know that's relevant to decision-making now about vaccines in this pandemic is observational. We don't have trials with the right variant, the right amount of time since it's waned. Everything we know about pretty much that influences our decisions is from observational studies, and I'll give some examples of that. But I think the idea of making policy without observational data, at least in this setting, is just nonsensical. We all have to do it because there's nothing else. And some of it is very high quality. So I look forward to continuing that part of the discussion. Just some disclosures and mentioning that although I'm now working at the CDC part-time on an IPA where they rent me from Harvard, I am speaking in my personal and academic capacity. So a couple of months before the pandemic started, Mauricio Santillana and I published this diagram, which was adapted from a more primitive one that we had published some years back, about the notion that there are decisions that public health authorities need to make and that individuals need to make, but in this case, public health authorities, and that the building blocks of those decisions should be evidence. And those are decisions like, how big should our response be? How should we choose who gets vaccines and other countermeasures first? What is the purpose of those countermeasures? Is it to prevent transmission, to prevent consequences, or both, and so forth? And those kinds of evidence are built up from building blocks of surveillance inputs, which may be clinical trial, clinical case series, maybe investigations of outbreaks, maybe syndromic surveillance, maybe case counts. In more advanced countries like the UK, it might be random samples of the population that are specifically designed for the purpose, and I'll talk about that more. But these are all inputs, and in a rational world, which we just heard in the discussion, ways in which we're not that, but in a rational world, we would design our surveillance systems to be fit for purpose to answer these kinds of questions, to build the evidence that would answer decision-relevant questions. So while that is not what we actually do to a large degree, it is nonetheless a useful regulatory or regulative way of thinking about it, normative way of thinking about it that helps us, I think, do better. What we've done in COVID in the United States and in most other countries, other than Luxembourg and the UK, I don't know of very many exceptions, is to count cases as our primary way of knowing how many people there are. And Stefan Barrel, who I don't know if he's here or online, but wrote a nice paper that I'm anxious to read that makes, I think, some of these same points based on my quick look at the abstract, about the losses of what we can't learn because of our surveillance systems not having adequate design. But if you think about what we're trying to measure, which is the incidence and perhaps duration of and prevalence of infection, and then what we actually measure, which is at the bottom of this funnel, these reported cases, there are all sorts of barriers and determinants of whether people get tested, whether they can get tested, whether they want to get tested, whether there are swabs available, and whether the test gives the right answer at the right time when they happen to take it. And so what we're measuring is this very socially determined and changing view of a biological and natural quantity. And that's the fundamental issue that I think has been a challenge in many surveillance settings in the past, but especially dramatic in this case. So I want to sort of make the case for surveillance as what should be really a set of different instruments. A lot of people really have one view of surveillance, that it's about genomes in some worlds, or it's about wastewater, or it's about counting cases, or it's about something else. And the issue is that really it's probably can be all of those, but those are means to particular ends. And we actually want tools for surveillance that are designed for those specific ends. So it's not, we don't want a single blade on our knife. We want multiple approaches. And I think you can divide the purposes of surveillance in various ways. I would suggest that some of the most important for categories are to detect new threats, new to a geographic area or new to the world, to assess individuals and treat them, which is, you could argue if that's really about surveillance or if that's something else, to characterize the severity of an infection and the effects of countermeasures, and to quantify the burden and how that's changing over time. And these are all really distinctive purposes. So I'm not going to go methodically through all of those in order to avoid boring everyone, but I do want to sort of give a couple of examples of those purposes and why and how we might approach them. So detecting a new threat, I think, is in many circles what people think surveillance is specifically designed for. And once you've detected it, it's done its job. I'll argue that that's not true, but it is an important purpose. And one reason to do that is to activate our development of countermeasures. You can't develop countermeasures until you know you have a problem. At least you can't fully develop them. Organizations that are trying to accelerate that development are, of course, doing preparation and trying to get countermeasures up to a point where we could fully develop them when a threat emerges. But once the threat has emerged, then we can begin to test potential countermeasures and evaluate their suitability. More recently, preparing for the spread of variants has become the primary detection goal for COVID-19 surveillance. So how do we design a system that's well suited to that? Well, geographic scale is important, obviously. It was convenient for this point that the first 4 critical variants of COVID-19 were found on 4 different continents, and didn't have to be that way, but that's how it worked out. And obviously, having surveillance in many places, and arguably also at airports in order to catch many places in an efficient fashion, is crucial. Size is also important. Bigger is certainly better, but not all the way to infinitely large. We don't have to do 10,000 samples in a short period of time if we're trying to find something at a particular frequency. And one of these things that you can publish once a decade or so, and we got the 2019 publication of this, but it was published in the 70s and the 80s, and is the rule of 3, which just says if you want to see something at a frequency of 1%, you need to measure 300. You want to see something at 0.1%, you need to look at 3,000. There's a very simple rule, and you can make your surveillance systems sized in order to pick things up at a particular frequency. What about quantifying burden? I think that's been perhaps the most difficult thing to do, especially more recently, especially in the beginning when testing was impossible, and toward the more recent times when testing has been done at home and not reported. But it's of course very important to quantify burden and also relative burden in different groups of people, so that we understand the trends, so we can figure out how much population immunity there may be, tell if our control measures are doing anything. identify needs in particular groups and inequities. And I got involved a little bit early on with Luxembourg because I thought that a place with lots of money and not very many people would be an interesting place to think about what you could do with a sort of gold-plated, I would say Cadillac, but that's the wrong nationality, version of surveillance. And certainly before I appreciated how important this was, some people in Luxembourg had the good idea that they could just do random samples of their population. And so they ended up sampling something like 10% a week of their population with swabs. And they were able to, therefore, to make an estimate every week of how many people were infected. That was an early demonstration of a more general principle. And the inequities that you heard about in much more detail from Anna, I won't dwell on here, but just to say that one thing we did at least document pretty well in the United States was the extent of the inequities. So how do you quantify burden? Representativeness matters. For other things, it doesn't matter. For detection, you don't want representativeness. You want to find the people most likely to have it. For burden, you want representativeness. And so this random or quasi-random sample in Luxembourg and a similar thing that was done in the UK was really important. Also important is data completeness so that you have, so that you can understand who these people are. A more innovative approach that we came up with, actually Michael Mina came up with, and a few of us helped him implement, was the idea that viral load can help you to identify the trend. So in a growing epidemic, most infections are recent. Recent infections tend to have higher viral loads, and so the distribution of viral loads cross-sectionally in one measurement can tell you something about whether the epidemic is going up or down, because of the asymmetry in the way the viral kinetics work in this particular infection. So I think there's space for innovation. I'll talk about some other sort of ways to do things that that when you don't have perfect data. But it's been surprising to me how reluctant people are to use viral loads or CT values in this pandemic because people think of them as error prone. And they're the province of laboratorians, as they're sometimes called at CDC. I think that's a real missed opportunity, and be happy to talk about that. This was a previous, I mentioned Stefan's recent paper, but Natalie Dean had a nice commentary some months back about the value of random sampling, which I've already talked about. The best way to know how many people have an infection is to sample them at random from the population. And she was extolling the value of what the UK had done. And the UK really, at the beginning of the pandemic, set up, there was some kind of a weekend discussion. I don't know what weekend it was. I think it was in February of 2020, but I spent a few days there learning what they had done. And there was some very important weekend very early on where they said, we need to know how many people have this, and we're gonna set up studies to do it. And they set up two, one longitudinal study of families, of households, that's called the Office of National Statistics COVID Infection Survey, and one repeated cross-section of individuals called the UK REACT study. And these were able to elucidate enormous numbers of important questions from the prevalence of infection, which could give an estimate of incidence, cumulative incidence, because they also did antibody measurements. They could detect new variants because they sequenced. They could study vaccine effectiveness because they could link to vaccine records. They could look at symptom profile and how that changed. They could look to see whether there was protection against reinfection. They could measure equity. Very importantly, they were able to look at infection rates by group rather than just bad outcomes, which is or detected infection rates. And when your detection of infection is reasonably consistent because you have a decent random sample, then your ability to figure out where the inequities arise through the cascade is much more precise, and they really use that, at least to understand and to some extent to target their interventions. And infection fatality rate. Many people have asked, have pointed out that the UK has a National Health Service, and that clearly enhanced this. But it's remarkable, and this was when my co-director at the CDC center that I'm setting up, Dylan George, and I went to the UK to learn about this. We really pressed them on how much was this an NHS dependent thing, and how much was this something you could do, we could do if we chose to spend the money. And the record linkage was helpful for some of these specific questions, but the random sampling was done from census records in the ONS case, and did not require the National Health Service. So I think it's easy to sort of throw up your hands and say, we don't have a healthcare system in the US, so how are we going to do this? But I think we could actually do something much like it. Talking of sort of alternatives that are cheaper and easier to implement, Andrew Gelman and Leonard Covello and others have proposed to look at at swabs of people attending hospital for non-COVID reasons as a relatively random sample, or reweightably random sample. So it's not random, it's not a representative sample, but it's a sample of people who have little ability to say no. If they have to be in the hospital, they have to get their test. Whereas people who are volunteers for a survey may be able to, or can say no. So to some extent, they argue this is a more, once you reweight it for the fact that this is an older population and so forth, once you account for the known reasons why people are overrepresented in the hospital, they actually argue it's more representative than a survey where people have the opportunity to decline. So an interesting concept, which is perhaps more implementable in some settings. The general point that I want to make, without again going through all the individual examples, is that we have what are known as target product profiles for vaccines and for drugs and for treatments. We need to develop target product profiles for surveillance systems that say, what is it we're trying to do? And then what's important? And so, as I mentioned, for detection, scale and geographic scale and size are really important. For understanding transmission, you really need small but very, very detailed studies. And I didn't talk about that, but those are things like household studies, where you want very deep data, but very, very little, comparatively little of it. And so I think a lot more could be done to sort of prepare what it is how it is we're gonna respond based on what it is we're trying to do and how you do each of those things well. This was my picture of the world, at least, and I think many people's picture of the world before COVID, that there were certain things that you had to measure once, such as what is the sequence of the virus? How bad is it if you get it? When do you become infectious? When do you become non-infectious? When do you get symptoms? How well does the vaccine work? And so forth. And then there were the things that traditionally surveillance looks at, which is the geographically and temporally changing things like the number of cases, the number of hospitalizations, and so forth. But I think the biggest lesson for me of COVID is that actually all of this is surveillance. Every one of those things is a changing quantity. None of them, and mostly it's because the first one, virus sequence, is changing. Also because our immune responses are changing, which determine some things like viral load kinetics, transmissibility, and vaccine effectiveness. So every one of these things is not a research topic in the sense of generalizable universal research. It's a surveillance topic, and we approach these things somewhat differently. And I think it had to become a surveillance topic because we learned that these things were changing. And that gets to the point I made at the beginning, that that we don't have randomized data on most of these questions, but yet we have to make decisions about how long you should isolate people or how frequently you should boost based on the observational data. And I would like to call that observational data surveillance data in the broad sense of something that is constantly changing and needs to be measured in a regular, repeated fashion. That has some consequences because surveillance is subject to more limited human subjects protections. It still includes some, but it doesn't have the same IRB superstructure often. And so that makes it more easy to do quickly. And it also is a different mindset of we're not going to just know this and then use it for the rest of the pandemic. We have to have a system in place to keep measuring it. So the conclusions of what I've said so far are this idea that surveillance is really a set of different activities with different purposes. And some of those purposes require big studies, some of them require very detailed studies, and we should set up systems to let us do that. On this slide, I put in red at the bottom that many of these things on the left are things where integrated health systems, where payer and provider are the same, or something like that can be particularly valuable. And so in the second part, I want to talk about some ways in which integrated health systems can inform pandemic surveillance. And I'm going to use the term loosely because the examples I'm going to give, which are ones that I've been involved with different collaborators, some of them are true payer providers and some are insurers and some are others. Well, I guess those are the two things. But the value, but integrated true payer providers are arguably the most useful settings in which to do answer many of these questions. And the main reason is because they have longitudinal data on their individual members, which allows all sorts of improvements to the observational study design, which are listed here. And importantly, you can ask a lot of different questions in the same integrated health system. And I'll give an example of that toward the end. Or actually, I'll give a few examples of that. So I want to talk briefly first about some work on vaccine effectiveness that we did with the Clalit Research Institute in Israel with these collaborators. And just to sort of illustrate what was possible with very detailed longitudinal data from an HMO that represented about half of the 9 million people in the country of Israel, and which had been very heavily investing for a decade or two, a couple of decades in the infrastructure to do medical informatics. So this was the first of several large vaccine effectiveness studies that was done as a cohort. And this was a matched design where we took individuals who received the vaccine and matched them to people who had not yet received the vaccine over time, over the first several weeks of vaccination in Israel. And one of the important reasons to do an effectiveness study when you've just done a trial is that you can look at rare outcomes like hospitalization, severe hospitalization and deaths, which are not visible in a trial of reasonable size. And this was able to show substantial protection against each of these outcomes. It's worth saying a little bit about the approach because it really highlights what the kind of health system allows. So as I mentioned, it's a matched cohort design emulating a randomized trial with about 600 individuals per arm. The matching was extremely detailed with this list of matching criteria. And I'll explain why in a second. Importantly, it also allowed us to use a negative control outcome, meaning an outcome that we would expect not to be affected by the treatment of the vaccine. And that negative control outcome was protection against these, against infection, symptomatic infection during the first 12 days. And we knew that was not to be expected because the trial had shown that. So the trial provided a negative control outcome or a evidence that an outcome would not be affected if you designed your observational study properly. And we were able to use that to check the adequacy of matching. And then individuals who were not, who were in the control group could enter the treated group over time. In addition to being able to look at rare outcomes, it could also look at subgroups and see protection in, for example, individuals who had multiple comorbid conditions or were in a particularly older age group. The way that we used the negative control outcome was to see how much we had to match in order to make the two groups comparable. So these are the first, this is a blow up of the first 3 weeks. And again, the expectation is that if you were mimicking a randomized trial well, you would see no discernible difference between the vaccinated and unvaccinated groups in the first 12 days or so, which was what the Pfizer trial had shown. And when you match on only age and sex on the right, you begin to see divergence almost immediately. And when you match fully on all of those characteristics, it's obviously not quite perfect, but it's much more consistent that in the first 2 weeks or so, there's very little departure, and then those two curves cumulative incidence curves start to diverge. We then continued to do vaccine safety and sequelae of infection. So matching was done as in the same way as in the VE study, but now for 2 exposures, either vaccination or documented infection, with the outcome being various bad things that can happen as a result of one of the 2 of those. Although the 2 groups were not completely comparable because each was a separate study done in the same database population, you can see that the risk difference for infection was strongly positive for things like cardiac arrhythmia and acute kidney injury. The sort of positive control for the vaccine was lymphadenopathy, the big blue bar in the middle, where you did get that outcome as a mild adverse event. And then you can see a signal of a couple of other things as well. With a database like that, you can also look at households because households share the health organization, this health provider organization. And so we were able to compare households in which we were able to compare outcomes in children who were not yet eligible for vaccination in households with 1 vaccinated parent, 2 vaccinated parents, or no vaccinated parents, and show a reduction in the parents' risk of getting infected, and then a further reduction in the risk of transmission if the parents got infected, or sorry, of secondary cases in the children. We couldn't prove transmission, as well as an overall effect of the parents being vaccinated on the children. I'll talk briefly about this. I'm going to skip some of the details just for time reasons, but United Health is not an HMO. It's as you know, a large healthcare insurer with some provider aspects, but mainly an insurer. And so this was an insurance-based study to look at sequelae and something like long COVID. So again, this took advantage of the fact of having longitudinal data on individuals and matched individuals who had become diagnosed with COVID-19 in the first year or so, almost year, against 3 control groups, those who had not become detectably infected with COVID, those individuals other individuals a year prior when there was no COVID, to adjust, to see what the differences in health-seeking behavior might be in the middle of a pandemic. And then comparators from prior years who had other viral lower respiratory infections or something that could be plausibly seen as that. And then we counted new events for the 4 months following the 21-day acute period and calculated a risk difference. And again, the point is not to see all of the details, although I'm happy to talk about those, but just to say that you could ask about a lot of different outcomes and the difference between individuals who had COVID and those who didn't in this population because of the longitudinal aspect. And interestingly, for different outcomes, the comparison to different groups was quite different. So on the left-hand side, with hypercoagulability outcomes, the SARS-CoV-2 infection group was higher than any of the comparator groups. For example, higher than other viral lower respiratory infections, and much higher than people who had not detectably had an infection. Whereas on the right, with cardiomyopathy, it was very much a typical lower respiratory viral infection. So those two groups were almost identical, but the comparator groups that didn't have infections were different. So you can ask more subtle questions that way. And you can look in subgroups, and we also repeated the analysis in their Medicare population, where you could then look at people among the over 65 by age group and by whether they had been hospital admitted. And of course, there were more sequelae in those who had been hospitalized. And then the last two examples I want to give are work that I did more or participated in, I did not lead, more recently in my CDC role, where we worked with Kaiser Permanente Southern California and Joe Lunard at UC Berkeley to look at the comparative severity of Omicron and Delta. So in late November, everyone was worrying about the Omicron wave that was about to come. We knew there would be a lot of cases, but we didn't know whether they would be as severe or more severe or less severe than the Delta cases. And so the big question in trying to understand the magnitude of the threat was about severity. And so over a several week period in December and January, the team at Kaiser Southern California and Joe at UC Berkeley managed to do a very rapid analysis of the data. And I think different people have different ideas about what rapid is. I think from CDC's perspective, this is rapid. And in fact, the period where Omicron was becoming dominant really began in January. Well, it was already dominant by January. It was becoming dominant in December. So this really was about as fast as you could do an analysis to get through the period of overlap, which was very important because you want to know on a given day, if someone is diagnosed with Omicron, what are their chances of bad outcomes compared to someone diagnosed on the same day with Delta. So the data were individuals who were tested in an outpatient setting using this particular testing approach, testing machine that Kaiser Permanente Southern California tended to use, which by lucky chance was able to distinguish Delta from Omicron by the S gene target failure, the means by which the SARS-CoV-2 has been trolling all the genome sequencers, because you don't need genome sequencing to tell which variant it is. It keeps on changing back and forth this S gene target failure thing. So if you use the right testing machinery, namely this single one from Thermo Fisher, you don't actually need to sequence the genome to get a pretty good guess of what variant you're dealing with. And we all find that very humorous. So you could tell Delta from BA1, which was the original Omicron, and then BA1, the original Omicron, from BA2, its successor, because they flipped this particular phenotype. And so in this slide, it shows the cumulative curves for the proportion of people on the top with Delta versus Omicron, Delta in red and Omicron in blue, that following their positive test were admitted for COVID-like symptoms, were admitted to the ICU, were mechanically ventilated and died. And you can see a dramatically reduced severity of Omicron on a per patient basis. And then the lower panel compares old Omicron to less old Omicron, so the BA1 versus BA2, and you see no detectable difference between the severities of each of those. And you could also look at length of stay in the same way. So I think of this ability to do this study as incredible good luck. There was a lot of BA1 and Omicron in Southern California. There happened to be a payer provider with deep electronic health records and a linkage to the state vaccine registry that had been worked out already. There was an excellent analyst, Joe, who had a pre-existing relationship with them. They happened to be using a machine that could distinguish Omicron from Delta at the time of diagnosis, so you didn't have to link sequencing data to it, which there wasn't enough of to do. There was this weird biological fact that you could tell BA1 from Delta based on that PCR result. And importantly, there was a CDC contract to build on, so we could move the money to them in less than geologic time to get the work done. So in this new role at CDC, co-leading the Center for Forecasting and Outbreak Analytics, one of our goals is to try to harden these kinds of capacities so that we don't have to have such good luck, because this is not going to happen again. This was just really, really fortunate that it was possible. Part of the good luck was this fact that we didn't need genome sequencing. And I think epidemiologists have got to become more comfortable with genome sequences for pathogens. I'm struck by how few people in certain communities that I'm a part of see the value of linking a genome sequence to clinical outcome. But if you don't know the genome sequence, you don't know what virus you're studying. And if you only know the genome sequence, you don't know whether it's bad or not so bad. So you really have to have both, except in this weird case where you didn't. And Jonathan Grad and I said a long time ago that the genome sequence should become the next item on the last column on a line list. And I think that's really true. It doesn't have to be the whole 10,000 nucleotides, but at least a name of a variant or a classification of what it is you're looking at. That is still pretty much in its infancy in the United States. Again, the UK is way ahead of us on that. But it does prove that it's possible. And then the last example I want to mention, because it just was another use of this kind of longitudinal data, and was an incredibly simple design, but well suited to a very particular question, was about Paxlovid rebound. So we knew that Paxlovid rebound occurs. We wanted to know, we were asked by CDC leadership, can you estimate how often this matters? We know it can be virologically important, but is it having clinical consequences? And this was for a very simple reason, that the CDC's guidance on Paxlovid had the statement in it, Paxlovid, following Paxlovid treatment, rebound can occur. It didn't have a number, it didn't have a frequency, didn't say if it was bad, it just said this can occur. And so we were trying to provide some context to that. So this was the simplest possible study design. Took 5,000 people who had been given Paxlovid and asked how many of them ended up in the hospital in the next following that, or in the emergency room. And the answer was that if you add those two numbers together, it's less than 1%. And so while the virologic consequences may matter for transmission, this showed in a very simple way that the Paxlovid rebound is not a major clinical problem. It's if anything, a virologic problem. I've rarely had so much trouble getting a paper through clearance, or through, in this case, clearance, but CDC's equivalent of peer review, because everybody wanted a more complicated study. And there are interesting, more complicated studies to do. But it was an interesting lesson, both in the sort of epidemiologist attraction to complexity when sometimes simplicity is better, and also in the remarkable patience of Debbie Malden, the first author, in being mansplained to by at least two reviewers. And she responded with grace that I would not have had. Broader conclusions. I think these kinds of research databases from insurers or payer providers can really be a source of surveillance data in that broader sense that I am trying to advocate. You can emulate a randomized trial and observational data. Often you can perform subgroup analyses and gain scale, all of which are important values of this. We should therefore, in my view, try to move from a research approach that has a fairly high activation energy to a surveillance approach for all of these questions, not only for numbers of cases. And that in some cases, these data sets are not huge. They're, a few thousand individuals who who are relevant to your study, and maybe a few million people in the total population. And they are not perfectly representative. They don't include uninsured people, almost by definition. But for many questions where you can plausibly biologically extrapolate that Paxlovid rebound in Kaiser patients is going to be a lot like Paxlovid rebound in Qatar or Mozambique or the Eastern United States, it's really okay to have a population that is both not huge and geographically specific. I mentioned the need to link clinical databases to fast sequencing, and we need to find ways to do this all faster and without such activation energy. So these are some colleagues, mostly at CDC, who have been really important thought partners in the view that I've championed, along with some other colleagues and from funders, and I will stop.
So if we could get the speakers to the front, and you want to join them? Yeah. And they're going to bring Corey's here with mics. So we'll be all set. Okay. A little technical work needs to be done here. No, that's fine.
Just announced people.
All right, we're wired.
Deserves a vote of thanks.
Yes, thank you very much, Corey, for all the AV help. We very much appreciate it. Right now, we are willing to take questions from the audience for the panelists. There are 2 microphones, excuse me, in the auditorium. You can also enter your question in the chat box and we will read it to the panelists. And our first question from the chat box is, are we including, if we know, vulnerabilities based on access to vaccination if desired? And I think this was a question from when Dr. Diez-Roux was speaking.
Can you repeat the question, please?
This is, are we including vulnerabilities based on access to vaccination?
Well, certainly, there's been documentation based on inequities in uptake of vaccination that have, many drivers. So that's been well documented. I may not be understanding the question completely.
If the person who submitted that question would like to add it to fill it out a little bit more, I'd be happy to read it.
Was it fun? How long was it?
We had to go down at 12:45.
Oh, wow. Okay.
Everybody else came down at like 1:15.
Okay.
And then it was 1:15 until 2:30.
Oh, okay. That's pretty long. So you didn't have 4th period?
No,
Oh, okay.
I didn't have part of 3rd period.
Got it.
They're all muted.
To mute everybody
Yeah.
who's on. Yeah, there we go.
Okay. It's like it says they're all muted. I don't know what's happening.
So this is a question for the entire panel. I think that the pandemic has reminded us that academic institutions are not nimble, right? So there were lots of questions that many of us had initially and wanted to ask and answer. But by the time you get an NIH grant in, it's 1 year later before you can do anything, or if you get a CDC contract or do anything. And what have we understood from the past and what can we do going forward that would allow epidemiologists to ask questions in real time? I mean, other than serendipity that Marc sort of alluded to.
I'll start. I think you're absolutely right. And in some ways, academics are more nimble than government, but the government often is the funding. So that's the challenge. I think this concept of administrative preparedness is one that people talk about sometimes, don't do a very good job of implementing, but the idea of having mechanisms in place by which those kinds of studies can be funded. For example, the NSF has this thing called the rapid mechanism, whereby you can get small quantities of money. I think it's capped at $200,000 in a few weeks. And our center at CDC works with NSF and has parked some money at NSF to do that. Some things cost a lot more than $200,000, and so it's not a full solution. But I think we should all be advocating from both sides to have some mechanisms in place that can make that happen more quickly. I mean, the UK is a really interesting example, and I think there are advantages and disadvantages to having kind of the structure the UK has, where a small number of people can make a lot of big decisions very quickly about a lot of money. There are obvious ways that can be abused, but it is also a real advantage if those people are trying to do something useful, and they really did in a matter of a couple of months draw up a plan and implement it with two very, very large studies besides a whole bunch of other things. I mean, so I guess my only point is that nimbleness is not like impossible. It's just the US system, for some good reasons and some bad reasons, is not that nimble. But we could work on it.
I might take a response to a somewhat different question, which is the response of academic institutions to the pandemic. And I think the word research was used, but I think in terms of the capabilities we could bring to public health, some of that was, I mean, in the case of Colorado School of Public Health, we responded within a week to do things, to put together a team that worked directly with the state on the modeling and other data issues. And I think, you know, talking to colleagues around the country, many did that. It wasn't a question of money. It really was a question of trying to do what we could do, should do as quickly as possible. Now that led on to some arrangements to actually pay for what we were doing, but we did a lot of things for free that were very intensive for, you know, quite a while. And I probably the same for
Yeah,
US especially.
yeah, I think, you know, it's a really interesting question because I think on one hand, at least I saw in many, you know, my institution, but many institutions, a remarkable pivot that a lot of people changed what they were doing and started to focus on COVID, even though, you know, there was no specific funding for that. People found ways to do it. I think part of the challenge was that it was not, and maybe this is too not possible, but it was not coordinated, you know. So I think, you know, sometimes I think, you know, if we were able to harness all of that and all these people that were spending time doing their little thing and harness that in an organized fashion to collect data systematically across multiple places, we could have done amazing things. So a part of it is the organization, the coordination, I think, more than the ability or the willingness to pivot, or even the money, frankly, because people found ways to do it. So I don't know, it's a really good question.
Well, and Hopkins deserves credit, I mean, for putting together the data.
Right, that's
So,
that's,
for example,
that is
yeah, so-
absolutely, yeah, that's one great example, actually, yeah.
But I think, yeah, I agree about the scale. I think, you know, the early days of how severe is this and are there a bunch of infections we're not observing? It's a challenging thing to figure out no matter what. But if you have every little place doing 3,000 people or 300 people, not randomly chosen and with their own homebrewed assay, you're going to get chaotic answers and the lack of organization, or in some cases, nonsensical or even destructive answers. And yeah, I think you're right.
So great talks, great group, and challenging time. I think from a technological point of view and information point of view, this should be the glory days of epidemiology. And the Hopkins dashboard put epidemiology on everybody's TV screen. And yet Walensky just issued a really scathing report against her own agency, saying that the staff had become too academic. Burned for all of us, right? But and perhaps too focused on writing papers for journals. So my question is actually about the journal. And Enrique, you put up a slide that was a little scary when you talked about social media and the journal, and dabbling in social media on science is really tough these days. So it's a general question for the panel. But if you think about the frontline practitioner of epidemiology, if you think about the policymaker and you think about the communication challenge, I'd love to get the thoughts of the panel on how the journal can be more responsive to that frontline immediate need. It's I think very related to the first question about being more relevant and quicker and bringing the science in a credible way.
So if I can jump in really quick, we have a chat question that's related. So I thought I'd read it and you could kind of answer it all rolled together. It's about communication around pandemics. Experts are often not great communicators, and communication experts aren't necessarily the experts. This pandemic shined a light on how important SciComms are Further, social media and the rapid spread of mis and disinformation is a huge issue negatively impacting public health outcomes. So can you speak a little more on that as well?
So I can take it from the journal perspective. Science is communicated in very different ways. And today, social media plays a huge role on how things are communicated. So my perspective and some of the editorial board is that either you can participate or you can stay out. We stay out lots of times in the past and has been miscommunicated. So the only way to try to combat the misinformation is to be there. And maybe we cannot be as effective as we would like to be, but we gotta be there. We have to be good communicators. We have to engage, because those are the people that we are trying to improve their lives. So I think that the landscape is changing in such a way that people are reading all kinds of things, including our scientists. Discussion has to happen faster. You know, as I started, when I wrote my first paper, I wrote also a letter to the editor. It took maybe 5 months to get published, and then another 2 months to get the response. Today, we have to have discussions that are rapid, right? That they go back and forth. Social media is a great way to have those conversations. I think that we need to take advantage of the things that social media does well, and we need to take advantage of what the print version of the journal, or not the print version, the peer review version of the journal will do. And I will pivot with that question to ask the panel on the same thing, that the structures that we have, I think that are inherently slow. We wanted to do it slow and well. And so the peer review process is slow, the grants is slow, everything is in a slow, because we used to believe that science has to be in small steps, incremental. And I wonder the paradigm of same question is this tension between rapid publications in the pandemic, you know, without peer review and all the aspects of bringing out that data out, compared to the one that is going through a process that is peer review. And I wonder, what are your thoughts about in the world of pandemic, how things have changed your views, or what do you think that the role of those two systems of publications?
It's too big an issue to answer. I mean, realistically, and if you look at how information is coming out from our institutions during the pandemic, look, Harvard, Hopkins, et cetera, were positioning their experts to be out in the media, as were other institutions. Journals were, and other outlets and sources. So I mean, in some ways, and we have different streams of communication that reach different people by their nature. And there's different sorts of receptors out there. And I think if we sort of look at the landscape of people who are receiving information, they differ in how they get it and how they take it in and how they interpret it and what should be reaching them. I don't think, use Anna's word, I don't think we have a system in place that acknowledges all of this. We sort of try, we acknowledge it, but it's in a sense bigger than what it is we do. So I mean, I think it's certainly an area where there are lessons that we should learn. And of course, now we have this counter of misinformation and deliberate disinformation, which is distributed far more aggressively than it ever was. And so I think our challenges are bigger. There's going to be lots of reports and commentaries. There's a National Academies Committee forming on what is disinformation and misinformation, even trying to get a common set of terminology. So I think this question addresses a you know, sort of a host of issues that we've all grappled with, one piece here and one piece there at a time.
Yeah, I think, this is also connected to the previous question in some ways. I mean, in an ideal world during a pandemic, the way you get information is not by people just publishing their own little things and, getting them quickly on the internet. You have a coordinated government response that is guiding collection of information, leveraging multiple resources, including academic institutions, and, delivering solid information. But what we had was a chaotic response where everybody was just putting out their thing, and we were relying on a group here to do, oh, look what they found. Oh, what about that? It was all like that. And so I think that's not the way it should function. And I think, so one thing, during the response to the pandemic, it's a different situation. It's an emergency situation where information needs to be produced and distributed in a way that is completely different from the way in which science advances. And certainly, it has to leverage science, but I think it's two different goals, two different circumstances. We can talk about whether we're slow in science independently and the peer review system and all that, but expecting, the peer review system or science to respond to the pandemic is, I think, a sign of our lack of acknowledging that we need a coordinated response. I mean, this, as a society, this is why we have government. and yet, we saw what happened, right?
So I think on the sort of preprints and speed thing, I'm sort of struck that there are 3 different timescales. There's how fast the information flows, which is sometimes the fastest. There's how fast our knowledge of the truth is changing, and then there's how fast the truth is changing. And the pandemic, this is the same point I made earlier about surveillance. Everything is surveillance. The vaccine, how well the vaccine worked in March was not how well the vaccine worked in October of 2021. And so peer review can't keep up with that by itself, or can't easily keep up with that. So I think when the actual facts are changing faster and the science is trying to keep up with it, then I think peer review, that's another reason to think of it as surveillance rather than research. Peer review isn't the answer, and it is in the way to some extent. It's the other thought I have is, I think part of why social media is so powerful, and I understand this is a small thing, but is that scientists are fairly allergic in journals and in public health publications to saying, we're not quite sure, and the data don't allow us to conclude this with certainty, but our best guess is that Omicron is going to be a big deal, is going to spread widely in the US. We can't know that until it happens. Or our best guess is that the severity of infection is somewhere between this and that case infection fatality risk. And so I think I hadn't really thought about what journals can do to encourage responsible speculation or responsible summation of what people think the state of the world is, even when they can't be absolutely sure. I think in public health practice, one of the things we can do is to just put that up front and say, and we've been experimenting with that in the publications on monkeypox that our center at CDC has helped to put together, which are called, we've been calling technical reports, which is what the UK calls it, and we're imitating them to a large degree, where we have a qualitative risk assessment, and we say, these are the scenarios that could happen. These are what we think are more and less likely. Here's why we think that, and here's how further evidence would change our view of it. That's not exactly science in the traditional sense, but that's the gap that good and bad social media fill. And before, in much of COVID, the good and the bad parts of social media. We're trying to take all these numbers that public health was putting out, often without any context or interpretation, and tell you what to think about it. And I think some of it is we can recapture that role. We are actually, as scientists, the ones best positioned to understand what scientific data do and don't tell us.
So Mark, surveillance.
Yeah.
I've argued in the case of tobacco that our surveillance has really been inadequate because we haven't captured these immediate trends of jewels surfacing or whatever else. But don't we need surveillance of social media for what the trends of information and misinformation are, so that we can, if things are going in the wrong direction for too many people, intervene? And you can put on your CDC half hat. And is this a role for CDC to be tracking what people are thinking or being told? Because that is just as important to prevention as everything else we do. I mean, that's certainly one clear lesson from the COVID so far and from past experience as well. So do we need some sort of systematic surveillance of the themes in social media as they emerge, come and go, and perhaps respond? And I probably, that would be with CDC if we, as a function of that agency, I guess, I'm not sure, but.
Yeah, I'm not sure either. And I can't speak obviously for the collective, but yeah, I mean, it is clearly it's a determinant of health behavior and of health. So I think it's an important thing to be monitoring. How to do that, I need to think more about, but yeah.
You have a question?
Yes. Hi, everyone. My name is Tom. I'm a second-year PhD student in ID-Epi. So just to say that I really appreciated all of the talks today. And my question is, it was alluded to that AJE needs to focus on diseases that affect the general public or have high impact. What do you think about studies about monkeypox or other diseases primarily affecting subpopulations, particularly MSM?
You went too fast for me.
There are questions about monkeypox and diseases affecting particular populations, particularly MSM.
If we are interested in those.
Yeah, and your role and your view of the.
Oh, yeah. And first of all, it has to be impactful. So if a paper is well done, there's no topic of limit. It has to be well done and good method. So send it in.
Thank you.
We have another question from the chat. Up to the first year or so, the COVID deaths per 100,000 was higher in metro areas than non-metro areas. But after that, the trend reversed. By July 1st, 2022, the stats were approximately 292 deaths per 1,000 in metro areas versus approximately 393 deaths per 100,000 in non-metro areas. Any thoughts on what might be the possible local public health policies or other reasons behind this kind of pattern?
I would, well, I'll practice what I preach. I don't know, but but an evidence-informed speculation is that vaccination uptake varied dramatically in different places and was very, very protective against dying from COVID. So that would be the first hypothesis I would look at, but whether it's the right hypothesis, I don't know.
I think, let me make sure I understand the comment. Was it that initially the more urban areas had the highest rates and then that pattern shifted to the rural areas? And I will say, Colorado has an enormous population along the Front Range and then many remote, rural and frontier counties. And we certainly saw that. And we were also tracking population mixing initially. And a lot of it was, along the Front Range, I think with less connections to the more rural area where there tended to be less mixing. And I think over time that protection just waned, I think, as more and more infections were seeded in these rural communities that were had somewhat more limited networks, which, is what we were looking at with some of the mobile phone data. And I think that also probably contributed over time.
Yeah, there was a really interesting study in Chile that the socioeconomic gradient flipped because Chile was late to get it, and it was mostly rich people who traveled to go skiing in Austria and stuff who brought it back. And so the rich neighborhoods of Santiago got it first, and then the bigger impact flipped over into the less wealthy ones.
I had a question that goes back to our earlier discussion about the next 100 years of the journal and then the next 100 years of the discipline. And John said, well, we don't have any more alchemists. One of the modern definitions of alchemy is someone who makes things better. So I think we might want more alchemists in the world. And I actually think epidemiology is really well positioned for coming decades. And that's because we tend to do many different things. We need to be able to talk to the data scientist. We need to be able to understand epidemiological methods. We have to know communities and social determinants. You need to know your disease. And I think that's important but at the same time, I hearken back to the impact factors and the transition to disease-focused dissemination of our research. So I just wanted to toss out about what are the implications of us having to do, team science is hard, but you need people who lead teams who can understand and speak the languages of the many different stakeholders around the table. So just throwing that out as a comment. And if there's any thoughts about two things, the direction of the journal, as well as sort of the impacts on training the next cadre of epidemiologists.
All right. So I think that as I said, I think that the view, I think that the journal is going to go where the field is going, which is, first of all, a much bigger integration of issues of social determinants of health. Clearly, we know that, but we haven't, it has a real focus right now. How is it all the exposure disease in the context of social factors, either macro factors or micro factors? I think that's going to be important in all aspects, because that makes it an impactful question. I started that saying we want a well-defined, impactful questions. And so because we study populations, then the structures and the social context will affect. I can't imagine any question that is not affected by social factors. So disease-specific exposure diseases in the context of social factors will be one that I see emerging. I get more and more papers of those that are cleverly done. I mean, there are so many really clever people out there that are looking at that. New study designs, I think we need to get more creative on how are we going to study 100 years of a lifespan into the career of scientists. Has been a difficulty, right? We want to study preconception exposures and how is that going to affect mortality long-term. I think that's an emerging area, both in the questions and also on designs. And then innovation happens all the time. So anything innovative will be welcome.
So I have to respond to Greg. I think I know who his advisor was. I mean, in part, my comment would be that the field is going to evolve. I don't care what it's called. It's going to evolve. And probably the most important thing that we should be teaching is how to stay up with where it's going. And, I showed you an HP programmable calculator from long ago. I think we have better tools now, and I think I know how to work with people who use them. And there are folks around this room who have been at their careers a long time who have stayed up in a functional way with where things are. And to me, that's what is really critical, because things are going to change a lot, whether it's new study designs, bigger data, superomics, whatever it is, things are going to change. And I think the educational programs, and I don't think we do this well. We have to pick the right people, but I don't think we really sort of help people learn how to learn for a lifetime, which is, I think, really critical and something we should probably do better. And the changes could be more rapid. I think that is predictable for the field.
Yeah, I think already answered. My question was related to that about education and thinking about the next generation and how the pandemic impacts how we think about teaching epidemiology and the core components. So John, I agree with you completely about teaching that. That's difficult to teach. But even I can say a few years ago when I was a student here, there were more infectious disease epidemiology components that were part of the core curriculum, you know, just basic outbreak investigation, surveillance. And some of that's gone away a little bit as we've become super specialized, and all of that is just in the infectious disease curriculum. Do we need to rethink, you know, what components we incorporate, particularly related to surveillance? Is it strong enough in our current curriculum?
I think, you know, there's this field out there also, which is called population health science, right? Which is this interdisciplinary approach to population health, right? That engages epidemiologists, many disciplines. I remember several years ago, someone who was very active in trying to develop that there's now an International Association for Population Health Sciences was saying, well, we want to do this and that and interdisciplinary. And I said, well, you know, that already exists. It's called epidemiology. She didn't really like that comment very much. But I think it's really important, I think, for epidemiology not to become, and I agree with John, it doesn't really matter what it's called, but I think not to become, you know, excessively narrow and focused. And we have a little bit, I think this is changing, but we had at a point, it was all about, you know, cohort case control studies, and it was all of, you know, causal inference, and which is all very important. But I think, you know, we need to think about surveillance, and we need to think about interdisciplinarity and how that helps us understand health. And so I think we have to stay current with those big problems and do whatever we need to do to address those problems. I mean, there's some historical papers that maybe it was, I can't remember now, Mervyn Susser or Milton Terrace. I mean, they wrote about this stuff, you know, about how epidemiology can't be reduced to a methodology. It really needs to be about the substance of population health and the questions about what drives population health and what we can do as a society to improve population health. So I think we have to keep our eye on those questions and train our students so that they can use whatever method they need to use to address those questions.
Totally agree. And I think there's a long way to go.
Yeah.
And I think we shouldn't forget the MPH part of it, because
Yes.
that's the larger number, and the people who are, I mean, as someone with more of a biological background who came into public health, I still 20 something years later, can't believe you don't have to take any biology to get an MPH. That's just insane. It's just crazy. And I totally agree about the idea that it should be a set of kinds of questions and not, I mean, it's like the difference between economics and accounting.
Exactly.
I think some places have done a good job of transforming the approach from saying, these are our tools to these are our questions for which you might want to use these tools. But that seems good. And I'll say it again, because it really drives me nuts. I think we have to be able to use laboratory generated data in epidemiologic studies. And I can't believe how much resistance in some quarters there is.
We're going to do another question from the chat. This question is to Mark. It says, wasn't the Spain ENE COVID nationwide population-based seroepidemiological study one of the remarkable attempts to get representative data from a population? Can it be an example for the US?
Sorry, what, the Spain?
It says Spain and then in parentheses ENE-COVID.
Yeah, the seroepidemiologic study. Yes, yes. So when I said the only two countries I knew about that did it, I was referring to biologic studies, but yes, more places did careful serologic studies, but not many more. And Spain was one of the leaders for sure.
Yeah, I had a question regarding rapid communication of evidence for the common people. I'm from India and currently still epidemiology student here. So during COVID pandemic, what I observed, of course, it was unprecedented situation. And recent years, our people had access to this technology, WhatsApp, all things have become common. And the topics of discussion were mostly political, but because of the pandemic, the infectious disease, all those kind of things came to the center. So they had access to all the, all the evidences which were coming. And as the situation was evolving, the evidence and the guidelines kept changing. So this also made people believe that, nobody knows anything. So even some illiterate people also, with whom I had discussions, they were also saying that epidemiologists, they themselves don't know anything. But how can we convince them that situation is evolving and whatever the thing was changing, it was evidence-based. Also, because of this technology, people had access to evidences from other countries as well. And most of the time, evidences should be context specific. But of course, they have access to all those things. So how to handle this kind of situation? Going ahead, also, because this kind of things will happen in future as well. Thank you.
That's a hard one. Yeah, hopefully everybody will comment because none of us are going to answer your question.
So we need to run out the clock.
I think so science evolves and, what we knew in March or April 2020 was what we knew then and it was replaced. And there were things that were said that were sort of turned back. A great example was our Surgeon General, Tony Fauci, and others saying, don't use respiratory protection because we need to preserve it for healthcare providers. Well, that was totally, in a sense, misinterpreted. I sacrificed one of my faculty members by sending him on to the Laura Ingraham show to talk about respiratory protection. And he survived barely. But I think things evolved. I think part of the problem, and there are really some points of confusion that I don't understand, and where there was not one voice in our quote authorities, and WHO and CDC I'll single out on the matter of airborne transmission, were somehow way behind where they should have been. And I think probably within a few months we should have said, gosh, this stuff spreads everywhere. This must be airborne. And for those of us who are washing down our grocery bags, I never did, but we had some very strange ideas of transmission. And I think it really took too long to bring that together. And I think that's the kind of thing that eroded trust of the public, because one week you're wiping everything down and suddenly maybe you don't have to wipe everything down and maybe you should be wearing respiratory protection when you weren't told that before. And I don't think and I'll single out really WHO and CDC, I don't think they did a good job of bringing the evidence together, reaching conclusions that they could stand behind. And fair enough, when the evidence isn't there, you still have to say something. And I think then goes back, and whoever said it, Marc or Anna, you still have to say things when the evidence is is not as definitive as you want. So I think we set ourselves up for confusing the public in some ways on critical issues. And that's only a part of your question. And there's the global context which you bring up and how information reverberates around the world. You guys aren't going to prize here.
Too hard a question.
So first, I want to thank everybody for wonderful talks. It was delightful to hear everybody's perspective on everything. So my question is, or question/comment is kind of a mix up between what Greg was saying about education and about Anna bringing in sort of the frameworks in terms of our questions, right? As epidemiologists, we're struggle more and more with what we teach, right? We only have so much room in our curriculum to teach, and we often focus on the methods, right? So that individuals, when they can get out, they can actually do some research and actually know what they're doing with the data. But it doesn't always say at perhaps the expense of having a good grounding in frameworks, right? And we see this also within the journal, right? In the sense that we see a lot of causal inference or methods and things like that on study design aspect. That is all great. So where in the journal's view of the journal, what place should it play in helping educate the frameworks? Because we are limited in what we can actually teach in such a limited time for our students.
So I guess let's sum up, right?
Pretty much you, Enrique.
That's a difficult question, and I will tell you why it's a difficult one. So number one is because I think that the journal has to play a role in education. There are some logistical problems with that and some ideological ones. The logistical one is that it's really hard for people to get to write educational pieces, right? So it's not that we get submissions, or even when we ask submissions on, can you do a tutorial on this topic? We have 100 people volunteering. It's cultivating those relationships to get people to write those pieces. And second is, the space of the journal, we are competing with many other things. So we have a section, it's called the Methods Corner. So we try to have something like 1,000 words on something that it's new, to give a general information to the public, to the scientific community, to get a sense of some new things. But overall, is a combination of both things. Brian, if you're willing to write the educational piece, please.
It depends on perhaps the educational piece, but so I do challenge the journal perhaps to open it up to printing a little bit more in terms of frameworks and getting away from the methods per se. I mean, methods are important, but we do a lot of methods teaching. And so, and there's no way that I'm going to be one to say that methods aren't important. But I will say that I think there's space for short articles, as you will, for broad overviews of frameworks, or perhaps even some biology, right? But I also think there's space online, right? We have online capacity to actually put out things that if our students and trainees are going to the journal, they can actually get a perspective of what an epidemiologist should be thinking about.
I completely, so to second that, so we have, I know that we have a podcast, it's called Casual Inference, and where all different things are being discussed. And the idea there is to be more of an educational activity. And then we have tutorials. So tutorials is a way to put up discussions about, educational discussions about different papers and different topics. But, we're going to combat misinformation by education as well. So it's our role to be participants in all aspects. And so the journal can try to promote, but every journal is on the willingness of the community to do this altruistic activity, right? It's all altruistic. People are reviewing for free, are doing all kinds of things, including, we have to include into that the educational piece. And so I definitely will welcome that expansion in any way that we can.
So I have a comment. I would actually bounce this back to you. And it's notable in epidemiology that there's plenty of good introductory books. And then after that, there's very little. And it's always, and I've made this contrast to books in biostatistics where there's plenty of, more elegant advanced methods books. So here we have one of the largest departments of epidemiology in the world with a curriculum that brings methods and has evolved, I hope, over the time since I left. And maybe it's time for you to take the lead in developing what should be sort of the forward-looking methodology curriculum and keep updating and put it in a form that would be useful for everybody. And so I throw that challenge, back out to the Department of Epidemiology and those who are leading the series. I mean, I think it's time to do that. You've had enough years now. I think if the 750 series started 2006, 6. If it started in 2006, you've got a lot of years of experience and time to bring things together in a coherent package. So there's something for you to do.
So with John giving us all more work to do in the next 10 years, I think it's a good point to finalize the wonderful day. I want to thank the speakers and David and the organizers for a wonderful, wonderful day. And please keep sending good stuff to the journal so we celebrate the 200.
Thank you.
Yeah,
I love my next questions.
Everybody here is invited into Finestone for a reception. And then you can have your Friday afternoon back. But please join us. Thank you.
Never going to be enough.
All the stuff is over here. We're just gonna make things confusing.